to 100. That looks successful.
Got him. Or her.
I don't know if that one was successful or not, but. All right, go ahead and zero it.
Do I want to rotate now?
Yep, let's go ahead and rotate. Back to flush. Okay.
All right, so
There
now
was
we got
at least one, maybe two more.
we got at least 2 shrimp, right?
Mm-hmm.
Yeah, at least 2 shrimp. I think
we
3.
have. I think we have at least 3, if not 4.
Okay, that's great.
But I would say at least 3.
That's a great start. We don't need any more right now. Let's try and get a few of those loose whelks in the foreground if they're easy to
Sure, we can get some whelks.
to get. We can put it in the
Do you want to put them in the same
Yes.
jar?
Yes.
Okay.
Whelk.
So welcome to the octopus garden. These are breeding octopus.
All right, go back to 1.
Many of them are on eggs, which we do not know how long they incubate for, but we've reason to
bump
expect
up to
based
40?
on similar species
Yeah,
that
do
it could
40.
be at
That's a
least
good
a year
start.
or more. Other follow-up questions: when we share our samples out to the world, you do not have to pay as
50.
a scientist. I mean, you may have to
That one's
ship
in.
them to yourself, but this data is available to the scientific community. You can rewatch our dives, can use our multi-beam mapping data, can request samples from these national libraries, and it's all available out to other researchers. So check out our Scientist Ashore program.
There's
And
at least those 2. Let's. All right,
Science
Aaron, let's
tab
push in some.
to get more involved there.
Let me bring it. Oh, you're all the way over. Okay. All right, go ahead and zero it.
And go back to flush.
No, now you can go back to running again.
50.
Yep. Okay, we got at least another 4 or 5 whelks.
Yeah, if we get 10 from this site, that's good. So we're at like 8 or something right now. Maybe a couple more. And
Let's zero it.
that would be
Zero.
fine.
Had a rock in the tube.
Back up, or you go?
Yep, go back to 50.
Yep. 50.
Oh, that was one of those tube worms, but it did not get sampled. Are you smelling anemones? Oh, that's totally different.
There's that big one.
That's 0. Let me get rid of this rock.
Nice. Okay. Back up to 50.
Yep. Aaron, can you brighten us up? Thank you.
Buckle up, Welk, you're going for a ride. So how do you make sure they don't swim out? You may have heard Paul talking about rotating them. There's one sort of intake that comes from the vacuum hose in, and then there's a carousel of canisters they can go into, so you can rotate them on a drum. when we're done vacuuming, we rotate it back to the flush jar, which is just kind of a generic one that we can use to make sure nothing gets stuck in the tube. But it also means by rotating away from the actual sample jar that we're on right now, that doesn't leave an opening back out to the tube from that jar. So it effectively kind of caps it off, seals it off as we rotate it around on the carousel.
All right, I think we got enough for now. This
Yeah,
is great.
I think we have a pretty good amount. All right, let's zero everything and rotate back to flush.
So are we calling that the end of that sample?
Yep.
Yeah, we can call the end of that sample.
Erin, a question for you. When Greg asks you to brighten it, are you adjusting the lights or are you adjusting the camera?
I'm adjusting the camera, so I'm opening up the iris.
Do you have control to adjust the lights?
No, so the ROV pilots control their lights.
All right, full wide, Aaron.
All right, so in the interest of time and eventually getting into those transects, we have one more specific site to check out, and that is the original octopus garden that we discovered with Hercules last October. So we can go ahead and close up everything and head on over there.
We are all closed up.
Do we want to still be running the pop sampler?
How far is that site from here?
Yeah, how far is the site? Do we know?
I think it's a couple hundred meters.
A couple hundred meters,
yeah,
and we're going to be up off the rocks, right?
Yeah.
Yeah, we can keep it running.
Yeah, keep it running. Yeah.
Okay.
And
Yep.
Dane, I
164
presume where
meters.
we may land over there, there's really only one place we found to land that's similar to this.
Mm-hmm.
It's very similar in terms of just
Yeah.
basalt, not a lot of kick up
Keep
in
it
terms of
going.
sediment. So.
Yep.
Okay.
All right, and let's come full wide on Argus, just so that way we know what we're dealing with tether-wise.
Yeah, let me just guess where Argus is here. So you're looking at that way and you see that. So you're probably here. So 190 is what we're gonna go.
Okay, so let's meet at 190.
There'll be just a couple meters of up and then down. And let me know when you're ready for that move.
We're ready for the move to be called in.
Okay.
It'll be a little while
Bridge
until it gets to us.
Can we have a step 50 meters bearing 190? Yeah, thank you.
So, Greg, you were communicating with the bridge then, right?
I was Rennie.
Oh, Rennie.
Rennie
Sorry.
communicates with the bridge.
And yes, I was.
Yep.
Do we want to check out the roaming octopus?
I was about to say, if you can come down just a little bit, we can
Yeah.
zoom in and see if we can see that third arm, which is on the right side, so we might be able to see if it's modified or not.
Aaron, get it. I guess would be a female then, right?
Okay, so you first count from the one by the leg, right? So the third one is just still behind the brittle star?
So, Chad, you're thinking that one is female? No,
No.
we can't hear you.
Yes, confirmed as female.
Oh, okay. All right, full wide.
I love the way they reach out, kind of charge those first front arms in front.
Renee, do you think on our last dive, the last octopus we saw was this species? I mean, like similar in appearance and color?
I don't remember what
It
dive.
waved goodbye to us
It
last
waved
dive
at
on
us.
the
Oh yeah, the one at the last. I don't know. I don't.
slightly different color.
It could be slightly different color. I have to look at that.
Could we get a zoom on that fish?
Sure.
Or it might be out of view now.
Jamie likes to wait until it gets challenged. All right,
Just
Aaron, push
trying to
in.
keep your job interesting. I thought the octopus on the
Is that a
manipulator arm wasn't
sablefish?
interesting enough.
Right.
Cuskeel.
Oh,
Yes, it's a
Cuskeel.
Cuskeel.
Okay.
Mm-hmm.
It appears that our USBLs.
All right, pull wide.
Just, we're not over there.
No?
No.
No?
I don't trust the ROV nav right now.
No.
Can't trust nav.
Okay. Never can.
And you're the one telling them not to listen to me. I'm just pointing that out.
Yeah, Paul, the moral of the story is don't listen to either one of us. Make your own judgment.
And that's
It's all on you, Paul.
It's
Yeah.
terrifying because I'm new.
Paul, how do you work this thing?
As we pass over this vase sponge here,
Yep.
Amanda's probably screaming at me, calling it something wrong, but sometimes we'll find interesting critters inside. Skate egg cases,
Right.
fishes.
Yeah, we may not have the angle to look
We may
down
not have the
in
angle,
it
yeah.
with anything except bubble, so.
No problem.
But it's amazing to see that one octopus in that little like puka, right? Tucked
Yeah,
in that
the little
little like ledge cave, you know? So you know that they're using all kinds of this habitat.
So let's look down with bubble, and we don't see anything inside.
Mm-hmm.
It's actually a rather narrow opening.
Bridge nav.
Kind of a squished face.
Let's add one more step. Thank you.
Awesome to have Science Communication Fellow Alex
right,
Havens,
full wide.
one of our alums with us. Thanks for tuning in from Alaska, Alex.
Oh, I remember Alex. I
Mm-hmm.
forget what cruise it was on, but
Ocean Networks Canada.
that would explain why I've tried to block most of it out.
We love our partners.
We love all our Nautilus partners.
ONC is a wonderful experience, and it is
Challenging,
very
yeah.
challenging.
but exhilarating.
Yeah. Like, are you ready to take it to the next level?
Oh man.
What if we introduce things to get tangled to on the seafloor? And other ships.
And other ships, yep.
Let's plug into the seafloor.
Ah, wild.
Pull up a meter or two.
Okay. Now that's a large, there's some larger squat lobsters, but these are white compared to the orange one that we sampled.
So
Video,
just
let's push
judging
in.
by where the ship is and where the vehicles are,
We're almost there.
well, we will not be able to stop without moving the ship back on this transit over. So
Wow,
just keep
that
that
is
in mind.
a
What is that
giant lobster.
thing down in the? Is that a?
That's one of those worms.
Is it?
They're called
Eggfish?
spoon worms
Oh, spoonworm. Okay.
on the last cruise, but I don't know if that's correct because Jan told me that, and she said she didn't know if it was correct.
Whisper down the lane.
Oh,
But a lot of diversity there.
decent
Look
size
at all the
lobster.
different creatures.
hydroids, it looks like. Crinoids.
Yeah, crinoids.
Crinoids, brisinghids, brisingids. Yeah. there's
Leather
another one.
stars.
Is that a? What kind of worm is that?
Peanut worm.
Is it?
Doesn't it look like one?
Yeah, peanut slash acorn.
Video push in
It
all the
looks
way.
like, yeah, acorn, right?
There we go, acorn.
Is that the same organism?
Wow.
Or is it different names?
Mm-hmm.
I don't know.
What is it? Mike got a really good sample of an acorn worm.
Yeah, they don't tend to come up so hot.
Right. Yeah, look at those hydroids. Amazing.
Some black coral.
This is like the 13th expedition where it's just like everybody practice identifying all your animals. You're like, here we go.
Right, USBL's back to being believable.
Oh, how'd you do that?
Excellent.
I just thought about it. I clapped. If you just clap.
Click your heels.
Rennie,
Full
could you
wide.
think about me for a bit?
Yeah, that was it. That's why your pump's working.
Yeah.
Is that just for Herc or Herc and Argus?
Well,
Oh, is
Herc
that a sea
definitely is
star?
looking good.
Sorry,
it's
a sea
a
spider?
giant-
Argus
yeah,
looks pretty good
Pictogoniid.
too.
Rennie, your favorite?
Pycnogonid?
Yeah, look at the big one, right?
Oh.
Just to the left of the lasers.
Yep.
Yeah.
Let's just
Sitting right
Video,
there.
blast
let's
on
push in.
past that one.
Yay, sea spiders.
Oh, there's another acorn worm at the
Sea
foot of
spider
it.
for the
Wow.
win.
That's enormous.
That
Yeah.
is. Wow.
There's some lasers on him?
Aaron, let's come wide a little bit, and I'll bring the lasers down.
Wow.
Yeah, it's
Yeah,
big.
That's
It
a big
is
one,
like-
yep.
Eight, 10 inches across.
Yep,
That sea
yep.
spider.
Ooh, gives me the chills.
Exactly. Thank you. Everyone was getting
They're
saying
so-
that I was ridiculous for imagining that
fuel.
thing. Yeah.
Only if you only have to be afraid if they're like, if you're a sea anemone, Rennie.
That's
I
got
mean,
to
but
have
if
at
you-
least a 20 centimeter plus leg span.
Yeah.
Yeah, I would
Yeah.
say if you stretched out leg to leg, you'd probably get closer to 30, 40.
Mm-hmm.
Rennie brought up a really good point, though, which is if you woke up and there was one of those crawling on your bed,
And I
It'd
bring
be
up the
terrifying.
same point of if you woke up and any sea creature was at the foot of your bed, you would probably be terrified.
Well,
Yeah,
I don't
but
know. It might
this
be
one just
heaven.
seems more
you know?
mobile, you know.
They
A lot
move
would be
pretty slow.
incapacitated in the air.
Aaron, you want to push in
Yeah,
all
what's
the way?
that small little white
Oh, I
thing?
was going for this small
Oh,
little
yeah.
white thing.
What is that?
That is a sea star, just really kind of crushed up.
Squished in
Okay,
there.
what's mine?
Little octopus.
Hey, yeah, little octopus on the
Oh,
right.
is it? Or is
Oh,
it?
Yeah, it is.
yeah.
It is. Look at that.
This is
Mine was better, Greg.
potentially one of the transitionary ages, or it could be a different
Little
species.
peak.
All right, well, let's come full wide. Let me land. We'll
You
get a
dare
quick view.
slip
Yeah,
it?
so it'd definitely be worth coming down and taking a look at it.
So
Okay.
cute.
Okay, so we've just been saying
We
that
do dare slurp it. They're a
we are
challenge.
missing some of the life
Which one
stages.
was on that right?
We are way far ahead. I need to back up a lot if we're
Yeah.
interested in this
Video pushing
beyond
again.
a quick zoom.
There it is. Hold that. Trying to not land on another octopus. Okay. Aaron, give it all you got.
Is it just a brittle
it's
star?
It's a brittle star that
yeah.
fooled us.
It is.
Depressions
alarm.
that look like eyes.
Happy
all
Halloween.
dressed, yeah, all
Those
dressed up
shadows
for
look
Halloween.
like
All right, yours
eyeballs.
was
All right, full wide.
Oh, we were so excited.
Yours was better, Greg.
All right, well, brittle stars are cool too.
Yeah, it's not that
Yeah,
they're not
it's great.
exciting.
So we had just been saying that we were missing a lot of these life stages. We were so excited earlier to actually see some
Aaron, what's that bright yellow?
little
The
octopus.
anemones that
Anemones,
crawl up
yep.
on these stalks.
Yeah, break out of
Full
their
wide.
casings and
I'm going to
hatch.
push back in front, Rennie.
So
Yep. Thank you.
incredible to see some hatching itty bitty little octopus, and then when lots and
The
lots
ship is
of
held
about that
on.
I
I'm
can
gonna let
respond
you guys
50%
sort
target
out, and
gains.
then I'll try to head
I
over
can
to the
really
target.
go fast.
But
That is a big black
we
coral.
don't know where they go in between, so we're keeping our eyes peeled
Wow.
to see little octopus in between, or cleverly disguised brittle stars in between. Hello to the biology class in South Carolina. Thanks for tuning in. The lasers are 10 centimeters apart.
So why do we call tan objects black coral?
Has to do with their skeleton.
Yeah, the skeleton's black.
Ah.
Correct.
Yeah.
Skeletons are black.
So, which if they're not sedimented too much and you zoom in a lot, you can kind of get a sense that that's what's back there.
I
But
see.
usually the morphology, you can identify it at least pretty
Yeah.
easily to black coral without seeing that skeleton.
Right.
It is interesting that we don't have any views that have no octopus in them. There's sort of ones and twos.
Yeah, I don't think we've...
So are there small heat seeps where the solitary octopus are?
Well-
we have seen solitary ones with shimmering coming out and warmer water. In the past, we have seen them not associated when they're solitary with a shimmering water or heat source. I mean, the species doesn't require it, but they've found this place, and for some reason,
And
it's beneficial to them. At least we think it is.
Are those cucumbers?
Mm-hmm.
Yeah, holothuroids.
All right, Aaron, let's push in. Got a little bit of time for his good zoom.
Beautiful coral.
Yeah,
there.
wow.
Yeah, you can kind of see at the base behind the yellow that there's a
Black skeleton.
Black, but it's hard. It's hard to
Mhm.
say until the.
And then
so
I
these
also
are
wanted to see
filter
what.
feeders.
Ah, that's why that looked funny. Sea star and coral.
Yep.
Yeah.
And nice sea
A
cucumber.
little
Yeah.
cup coral.
Mm-hmm. So, sea cucumbers relative to the sea star.
Yep.
Same body plan, just sort of stretched out in a different shape. They are really important recyclers down here in the deep sea environment. They eat
A
little bits
floaty.
of marine snow and detritus out of the sediment and recycle it through their own bodies. Are the black things, are those sponges?
Those are those worms.
Oh, worms,
Yeah.
okay.
Is that a tunicate or another holothurian?
I don't know, can we get a shot of that?
Where? Oh,
Right
yeah.
below the whelk.
Yeah, yeah, right.
Yeah, hang on. Let me get in a little bit. Okay. Aaron, give it all you got, although I think you're already all the way in, aren't you?
Yeah, that's it, unfortunately.
So it's tiny. Well, let's get the lasers in the shot. Yeah. So that thing is very tiny.
Yep. It's hard to tell.
It looks like a tunicate because every now and then you get the iridescent.
Mm-hmm.
You talking to us, Jamie? Someone is calling from.
What is that thing on the very bottom there, that kind of gray?
In the middle now?
Yeah, in the middle now.
It's probably an encrusting sponge.
Oh.
Yeah, let's push all the way in.
Thank you, Amanda.
That's it.
So you're watching on that quad
All
view,
right,
the
let's
little
go.
red
Oh,
thing?
T-4, okay.
Is that what the other one was?
Yeah.
Yeah, I think that's what the other one was.
She said the other one might have been a xenoturbellid. Am I getting that right, Amanda?
I don't know. It kind of looked like this one.
Yeah.
Awesome.
Okay.
Yeah.
That's fantastic. So we are
All right, let's
near
come full
Davidson
wide. I'm
Seamount,
gonna
about 50 miles
go
offshore.
back out in front, and then we'll find something
Monterey,
neat to
California.
look at again.
So the ship's done moving, but Argus is still swinging. 'Cause
Yeah.
it's
It's very deep. Yeah.
So deep.
So
Sometimes ship moves can take 15 minutes to.
that was 100 meters I called in.
It's a big end.
So I'm going to call in another 50 or so once we start to see where the vehicles are settling.
How far are we still away from
Hercules is still 100 meters away from the target,
Bighorn? Okay, got it.
but the original distance was 160, so the ship has already traveled 100, but the vehicles have not yet.
Gotcha.
Catching up.
Catching up.
So we do get to sample for micro-communities and invertebrates
there's
when we take
always
sediment
a
samples.
octopus in screen.
Also, our environmental DNA can help detect the whole communities that are there. Depending on what their process, they can look specifically for different groups like corals and sponges, but it's a way of measuring an entire community based on isolating the
different
DNA
coral.
that is just in the water from sloughed off cells.
I'm
mucus,
going to
that
add
kind of
another
thing.
step in there. Bridge nav.
Aaron, let's push in.
Let's do one more step. Thank you.
Ooh.
That's a really pretty.
Ooh, what kind of coral is that?
Huh.
Is it a...
It's an octo coral.
Yeah,
Yeah,
I can't...
don't
Come
know the
full
genus
wide.
on
I
this
can
one.
spin around to get a better.
Yeah.
And little cucumbers.
Yeah, this is a great community right here.
gherkins.
Sea star.
Yeah, gherkins. Just right for pickling.
And look how big the purple one is off to the right.
Yeah.
It's a chunk.
This branching pattern doesn't look familiar, but
Exactly.
we've also Been all
All right,
over the remote
video, let's
Pacific, and
push
now
it.
I'm getting everything all mixed up.
I know.
Wow.
It sounds
Oh,
like
Is
oh
it?
no, that's a sponge. Yep.
I don't see any bamboo patterns. Does anyone else in the
No,
skeleton?
no bamboo pattern.
No bamboo.
Definitely
It is
an
an octocoral,
octocoral.
yeah.
Somebody at home is
But
like,
this
it's
is
this.
really delicate branching
Yeah.
pattern I haven't seen.
I wonder if it's a hydroid that's taken over a skeleton of something else.
No, I think that the
Usually
polyps
you don't,
look.
yeah.
The polyp
They
You
look.
don't
bodies are
get
pretty
that kind of
big.
spacing.
Yeah. Wow.
Since we're not over event, is this one of those places we wanted to take a Niskin?
Oh, as far as baseline?
Well, corals, there's black corals, there's this coral.
Oh, for eDNA.
Yeah.
Yeah.
But we're not over event specifically.
We don't need to fire one now. It's
Sure. Full wide.
So Amanda, can you tell us what that stalked sponge is? If you're listening.
it's lunchtime.
It's
Yeah.
lunchtime.
Yeah.
Everyone's
There's
getting ready.
another black coral
Oh man, is it
coming up. It
lunchtime
looks
already?
like. Not for us, Greg.
Well, yeah, but it means that the watch is almost over, and I'm kind of surprised by that.
So we have some people
See,
watching
all those lines are
who are
those
playing our
worms.
ROV bingo. They're watching Nautilus Live Bingo, and I have to say, you can't just send in the list of terms we're supposed to say. That's not how Nautilus Live Bingo works. But Greg, you just gave him one by saying we're getting ready for a watch change.
Yeah, I don't know that I know what's on Nautilus.
For a
Live
while,
Bingo.
like the free space, which is bridge nav. I mean, we didn't say that till the end of the watch today.
Yeah, there's a whole series of cards, and there are also blue water bingo cards and seafloor bingo cards to follow along and watch.
All
with
right,
us.
Aaron, give it all you got. So you can see the black underneath that one a lot better.
Yeah.
Yep. Yep.
So cool.
Aaron, you want to focus in the background on the other stalked thing? And we haven't seen any, is that a,
There's another little coral there.
yeah, but is that a,
The clear one?
yeah.
I don't know what that is. Creature. Yeah. Doesn't look like one either. Yeah.
Full wide.
Is that what that was?
Yeah.
What was it?
Carnivorous sponge?
Wow. Seafloor is weird things.
Alright, that ship move is complete. Should be positioning you where the octopus garden is.
Okay. Well, that'll be good. We'll turn them over at a new spot,
Yeah.
hopefully at the right spot.
That'll be like just like we had it in the beginning
All
of our
right,
watch.
we will check to see if this
There's
is
one
a
on the
male.
move.
A lot of black coral here.
Yeah.
Yeah, that third arm does look like it's exposed, so we probably
Video,
can
let's
check.
push in.
So the third arm would be kind of behind that right eye,
Okay.
second from the left, but he or she has it curled up pretty well. Can't see the tip of it.
Is it that part right there?
Yeah,
Yeah.
the tip of the arm is below the siphon right now
Yeah.
of arm number
Show us
3.
her arm.
So if this is indeed a female, are we to assume it's not in a brooding posture? So Probably.
Right, yeah. If it's a female, this is not in any kind of brooding posture.
Yeah.
But
It's a lot
just-
larger than the ones that were brooding.
Mhm.
Maybe I'm mistaken.
Well, maybe instead of being folded in half.
We saw a couple of females though earlier
Let's
that were
come full
fairly
wide.
large as well
Yeah.
that weren't in a breeding posture.
Out for a stroll on the seafloor. So we are checking to see when we can of whether or not your male or female is there. Just because we're interested in the ratio of the brooding
I don't know
octopus
that I
or
believe that
female.
last one.
There's probably a 50 to 1, 100 to 1
over here.
female to male ratio. So we're interested in seeing if there are males out
But
and about
I'd kind of believe the clusters.
looking for mates.
Yeah, I think it's doing good now.
I wonder if there's a bigger vent someplace that is changing the density just enough. Right, remember when we
You
were
mean in the
around,
water column?
when we were around PLB?
It could be that would change location, which we saw earlier, but it would not change the noise that we were seeing. But we did see it go way off to the east. That changes the sound speed through the water column, and it would make
Right.
it think it's somewhere else. But
Paul,
You're
you can
over.
come down to 25.
Okay. You're over a pretty large feature right now.
Yeah. Am I expecting to kind of drop off of something to get to the
yeah, you're
garden?
kind of like easing off. Like these are
Okay.
you're
So Chad, what do these octopus eat when they're not, not eating?
I
sitting
can
and
only
brooding?
guess based on a traditional octopus diet of shellfish and things of that nature. So crabs and lobsters, some of those little guys that we've been seeing crawling around, perhaps the whelks, even though we saw that one mom reject it. But of course, we're under the assumption they don't feed at all when they're brooding. So
Right. I will
may-
point out that there are no octopus in view right now.
there's Mike and future Mike.
a rarity.
Yeah, that is a rarity.
Yeah, watch. Mike and future Mike. Future Mike, Mike.
Yeah.
Yeah. So there's
I
lots
like
of questions
that.
coming in about what could we see this, could we see that? We never know what we'll see. Fish don't prescribe to our schedule, so
Yet.
yet.
We're working on that.
But certainly we've seen sharks, certainly we've seen crabs, we've seen all kinds of different animals down on the seafloor. You can check out some of those highlights on photos and videos. But
We have some feelers out about it.
a variety
Aaron,
of
let's
different
push in.
fish
We're going for
live
the
down
anemone.
here. And we just kind of have to keep making observations, the same thing that we are doing.
Oh, too much. Much too much.
So right now we are on our way over to the site, the
Dead
original
sponge.
Octopus Garden Discovery site. It's a couple football fields away from where we just were, so we're slowly making our way across the seafloor. Head over there, and we're just looking
far
at the biological community, so all the animals that live in this neighborhood that we can
Let's come
see. So
full
that's
wide.
a sea anemone and a sea cucumber, the large yellow fan-shaped organisms. That is an animal, and that's a black coral. They filter feed.
Stalked crinoid.
We have seen a few fish. Most fish broadcast spawn, even here down in the deep sea, so that's not something we see a lot. Lots of questions coming in about that, but not something we typically see. Sometimes we get
So
a
I'm assuming
predation
your
event,
positioning
sometimes
Argus
there's a
will kinda end
reproduction
up
event, but
off.
less common.
Well, currently it's just heading towards the point, but but judging by depth,
Octopus
we're going to
again.
have to go a bit deeper because it
Yeah.
says 3321 for the depth, and we're only at.
Wow.
That doesn't seem possible. We'd have to go way off of this feature. 3321. Yeah.
It looks like there's a bunch of octopus to the left.
Yeah.
Yeah, but
Yeah, this
They're
site
kinda
is
falling off
just
the
below
side.
the summit of this
Right.
feature.
Hmm.
And you'll start to see them in these linear cracks and crevices that traverse dozens of meters. And eventually buried in here is a pool of about 100 individuals where we landed and got some of those first striking images of a lot of that shimmering water coming out.
Do you happen to know where the depths came from that are in the dive plan targets? If they were from
These,
us or the Bathy or a different organization?
They came from Mbari's dives,
Okay, because they're...
I believe.
There's no way I can be that deep at that position.
Yeah, I'm seeing a discrepancy, in which case there could be an offset in the XY, or it could be that their depths are a bit different than what we're seeing.
But we're on the right path, is what it sounds
Yeah, I mean,
like.
this is starting to look like what we're headed towards, right?
So we just completed hour 3 of the persistent organic pollutant sampling. So we estimate we're up to 108
depth was
liters.
when we landed.
What's up?
Going for a target of 400.
Eventually.
So our Herc depth when we landed was only 3191, and that was right here at that vertical vent site. So we never hit these depths that are the Mbari targets.
Here goes one swimming away. Let's get a shot of this one swimming.
Aaron.
All right, so this looks like a female. No modified arm.
Gonna kill the lasers.
That's a great shot.
Yeah.
Yeah, we haven't seen this much movement in this area.
Yeah, I've seen a few swim, but usually not this high off the seafloor or this active.
Just going for a morning swim.
What a gorgeous animal.
Yep. Just amazing.
And we're getting a great view at those arms, so that is how we can say this is a female. On the third arm, there would be a modified different arm extension on males, and that's used to deposit sperm packets into the mantle of females. So when we have a nice, clear view of an animal like this, you can, just based on that external physiology, make that determination about male or female.
Paul, can you tell me what your Argus payout is right now, the read on the winch?
Yeah, 3151.
3151. Our depth says 3155. So I would trust the depths we're seeing over the depths from the Mbari targets.
The pool that we found at the garden is definitely over 3,200. I know that.
So as far down as I can follow it before I hit rock.
No problem.
No, it's a great shot.
Yeah, 32, I believe, but I don't think we
right,
could be at 3325.
Yeah, I think you're absolutely right. The garden did not cross 33.
Yeah,
I think you're right.
Not sure if there's what kind of depth sensor they're using or if it differs.
It could have been a transcription error too.
Yeah, or going by the bathy rather than the dives. Never know.
There's your jellyfish, Aaron.
Oh, look at that.
Oh, the scrubbing
Go
bubbles.
for it, video.
Scrubbing bubbles jelly.
Yeah.
Pedunculata is ringing a bell right now for that type of jelly.
Wow.
So cool.
Yeah.
Thruster wash.
How's
So
our
you
ship
can tell
position?
who all the teammates are by looking underneath
That's
the
probably where
question
Argus
feed.
will settle, somewhere around in there.
You'll see names,
So,
and if you click on the bios of every person on the team, as we change over to the watch group here in a few minutes, those bios will automatically refresh, or go ahead and refresh your browser to meet the 12 to 4 team who will take you for the next 4 hours.
All right, because I want to be downslope, I'm going to pull Argus off like 25 meters,
Sure.
30 meters to...
Chad, do you remember this big coral? Is anything that we can use for reference point?
Oh no, we find quite a few of these sticking out of
Okay.
this vertical slope.
I'm going to go 30 meters at 145.
But I would continue to head, yeah, southwest.
Yeah.
Bridge nav. One step, 30 meters, bearing 145.
It's incredible.
you.
Just look at the density, just ratchet up.
So that'll-
here.
Yeah, that'll keep us around this area, but pull Argus off the slope. So we can investigate a little bit better.
So do we not trust this marker for
I'm sorry. It's not that we don't trust the XY, it's just that the depth is.
Yeah, just don't trust the depth. It may, like I said, it may have been a transcription error. We're heading in the right direction.
Yeah, this is looking pretty good. This is looking like what we saw last year, right?
Yeah. There'll be this kind of 45-degree slope with all of these linear features with octopus lined up in it as we head southwest, and eventually we'll find a pool, that could be the one right there in the upper corner.
Wow.
Oh, it just came down a bit. Should I come down?
No, you're good.
Okay.
You can come down to 25. You think this is our pool?
Yeah, I think that's the pool. So that front basalt there is where we landed.
Okay.
Yep, this is the pool. So this is the area we first discovered last year. And this is the first place, well, I guess technically the second place we sat down and saw shimmering, but it was the first real big aggregation of these octopus. So this is it. So this is going to be an area as we transition from this shift to the next shift where we're going to be conducting some more oxygen and temperature measurements. We have one more temperature logger, and I figured we'll wait till we find another spot. And this would be a good spot because it's one of our go-tos that we could leave the other logger and potentially collect some more animals. But whenever you guys feel like it, you can settle down on the bench there and we can move forward.
Sounds good. I think your pilots are actually off headset as they're starting to hand over. So
they're handing over, yeah.
Mike will do it as soon as he's in.
Great.
Thanks everyone for your questions. Total Force here to carry you for the next 4 hours. Stay tuned. Obviously, a lot more to see.
See ya. Okay. Thanks, Greg. Roger.
Yeah, we're still getting situated back here, but we'll be ready soon in the science row.
Cool, thank you.
Good afternoon, everybody. This is Scott Simon from the Marine Science Institute at UCSB. I'm the current Science Communication Fellow on watch. If you're just joining us, we are at the Davidson Seamount in the Monterey Bay National Marine Sanctuary. In the Octopus's Garden, we're actually at the original site right now, exploring, doing some collecting. We'll be beginning our transects of the seamount later this afternoon. Excited to have you all on board.
Hello there.
Hello.
Hello, we're
Morning.
all transitioned over back here in the science area.
Okay.
So I think our first thing we're going to do is try and set down here at this area we call the Octopus Garden and look for shimmering water in areas where we can take some temperature and oxygen measurements and potentially leave one of the temperature loggers behind to record temperature over the next year or so.
Roger. So I just crept back and had a look. This is a nice place to land, so you want me to just get up as close to them as I can?
Yes. Especially in areas maybe where you see a lot of cleared rock, the black rocks, versus the sediment-covered rock.
How about this stuff that's in front of the porch here?
Where all the, you see all the whelks, that kind of thing?
Yeah, that's...
Or are you saying this kind of...
Oh, this kind of rock with the octopuses? in front of us? Is this what you want to get up
yeah,
to?
actually, so say the upper left
Yep.
area, just above that yellow rope.
Yep.
We should be able to reach them pretty easily, that area. For those of you who are watching who saw the video of our discovery of the octopus last year, you may recognize this location because it's featured prominently in that video. It's where we say, oh, I see a shimmer.
I don't know if we're saying that this time. Do we see a shimmer?
I thought I saw some. We had to zoom in.
Maybe in that, see that hole to the right below those two Brazinghids? That might be a good place to stick the probe.
I'm sorry, I'm still getting the voices. Anne or Jennifer, I think one of you maybe bring your mic a little closer.
It's probably me, yes?
Or is it me?
Well, you're both perfect now, so.
Oh, perfect.
Sweet.
Do you guys want to quickly introduce yourselves?
Oh, sure. I am Jennifer Brown. I am ecosystem scientist with Monterey Bay National Marine Sanctuary. I had the pleasure of being aboard Nautilus 2 other times, and I'd say the highlight was last year when we found these aggregations of octopus. So I'm happy to be back.
And my name is Annie Hartwell. I'm a graduate student at the University of New Hampshire, and I'm very happy to be on this cruise because I had the privilege to study a second cluster of these octopuses in a different part of the deep ocean.
Very cool. My job as the Science Communication Fellow is to communicate your questions, share them with the scientists and the ROV pilots on board to help guide you guys through this
a little bit of shimmer
experience together.
I don't really see much.
Yeah, do you see that hole just below those two Brazingid orange stars? Like
yeah, so
1 o'clock.
Maybe stick the probe in there.
Yeah, sure.
Thanks.
We've got a question. What does the shimmer show signs of?
Temperature differential. So the fluid that is coming out of the rock has been heated as it flowed below the crust on the bottom of the ocean, and now it's coming back out to the bottom water. And this is one of the discharge sites. And as it moves through the crust, it warms up, and then it comes out with a higher temperature. And we don't know about the other chemistry. We can't say how similar it is to the bottom water at this point. That's why we're here collecting samples to learn that great information.
Right, you might see a bunch of, to the bottom left of your screen, you see a black milk crate with a bunch of white and metal instruments in it, and those were used to collect water samples earlier in this dive for that very purpose.
Actually, we might try to fire the last one.
Do you know which
later?
one it is?
Yeah, I know which one it is.
Yeah, okay.
So we're just using the TDO temperature and
Yes.
okay.
That's our data logger.
You want to introduce yourself?
Oh yeah. This I'm Magda Flores. I'm the data logger on this watch. So my job is to record everything that we do and make observations and what we see on the seafloor.
Thank you, Magda. To my right down the line.
Sure, I'm Ashley.
Wow, these guys are everywhere.
I'll be conducting the video on this leg of the dive.
If we want
we could use the
we can use the Herc
Thank
probe
you,
to
Ashley.
start
Okay.
if we want.
just to get deep into the.
Yeah, and to let this octopus get a chance to relax and let
Make
go.
a decision.
Oh, wow, she's waking up.
Yeah, she's holding on.
Maybe
Maybe she'll want to live here.
Yeah.
Go join the garden.
She came from the vent, right?
even the other one looks like she's more active now, the one on the arm.
I hope that they might choose to depart.
Yeah.
I don't know, we could get them a little Nautilus hat, vest, and t-shirt. I think they do just fine on board.
Our lead scientist on this watch is Jennifer Brown.
Yes, that's right. I
Jennifer, we have a question.
Okay.
How are the octopus purple?
I'm full wide.
How are the octopus purple?
Oh.
Well, many octopus can change their coloration quite a bit. We have not seen this octopus do that. Now, they live in the deep sea, so usually they're in the dark. And changing color is probably not something that is very advantageous to them. So, you know, it may not be worthwhile for their bodies to invest in the camouflage that other shallower species can do. But in the deep sea, you often see animals that are white, gray, black, or red. And that's a form of camouflage because it's very dark down here and you can't see those colors in the deep sea. So that would be my thinking on that.
Hey, Aaron.
Very cool.
Can we step Argus like 10 meters forwards there? No, around. Yeah, just basically 10 meters towards Herc. We're going to be working here for a while, like 300.
Hey, Valeria, great to have you on board.
Thank you.
Again, we are in the Monterey Bay National Marine Sanctuary up off the coast of Monterey, about 50 miles off over the Davidson Seamount. These are the temperate waters of our oceans. I always like to say we have 3 flavors of seawater. One is temperate, one is tropical, and one is polar. These are temperate waters, so we wouldn't expect to find any seahorses
we're getting
here, really.
Kind of far away. We're going to start pulling pretty soon on Herc. But we'll still have another 30 meters off the wall, I think.
We've moved. We're getting pulled around.
I'm going to come down to 15 meters delta.
Yeah.
Jess, how's your day going?
My day's good. We're just on the seafloor.
Our viewers are asking how our day is so far.
Good so far. Just a little, lot of instrumentation work for us here, so that's exciting. So it's just to confirm, same as last night, we're just dropping targets as observation. Yep. Okay.
I have no mouse.
Jess is pretty busy right now, but Ashley, we have a question. Do you have any advice when submitting an application to join Nautilus?